Welcome to the BiochemSerye episode entitled, Overview of Carbohydrates. This is an episode of the playlist on chemistry of carbohydrates, which is linked above. Feel free to click and visit the playlist above to visit the other episodes on this topic. To start off, the carbohydrates are a group of naturally occurring carbonyl compounds, either aldehydes or ketones, that also contain several hydroxyl groups. The carbohydrates include single sugars, like monosaccharides, and their polymers, the oligosaccharides, and polysaccharides. Let us now look at the overview and generalities of carbohydrate chemistry. Let us begin with the introduction to carbohydrates. Polymeric carbohydrates, as well as some disaccharides, are important components of food. In the gut, they are broken down into monosaccharides and resorbed in this form. The form in which carbohydrates are distributed by the blood of vertebrates is glucose, commonly known as blood sugar. This is taken up by the cells and either broken down to obtain energy, like glycolysis, or converted into other metabolites. Several organs, particularly the liver and muscles, store glycogen as polymeric reserve carbohydrates. The glycogen molecules are covalently bound to a protein, glycogenin. Polysaccharides are used by many organisms as building materials. Oligomeric or polymeric carbohydrates are often covalently bound to lipids or proteins. The glycolipids and glycoproteins formed in this way are found, for example, in cell membranes. Glycoproteins also occur in the blood in solute form, exemplified by plasma proteins, and, as components of proteoglycans, form important constituents of the intercellular substance. Simple monosaccharides consist of a linear chain of 3 or more carbon atoms, one of which forms a carbonyl group through a double bond with oxygen. The other carbons of an unmodified monosaccharide contain hydroxyl groups, resulting in the general formula for an unmodified sugar of CnH2nON. The suffix ose is used for the names of sugars. If the carbonyl group is an aldehyde, the sugar is an aldose. Subsequently, if the carbonyl group is a ketone, the sugar is a ketose. Fructose is therefore a ketohexose, and glucose is an aldohexose. Monosaccharides are also classified according to their number of carbons. Sugars containing 3, 4, 5, 6, and 7 carbons are called trioses, tetroses, pentoses, hexoses, and heptoses, respectively. The most important natural monosaccharide, D-glucose, is an aliphatic aldehyde with 6 carbon atoms, 5 of which carry a hydroxyl group. Since carbon atoms 2 to 5 represent chiral centers, there are 15 other isomeric aldohexoses, in addition to D-glucose, although only a few of these are important in nature. If we recall, the most frequent cause of chiral behavior is the presence of an asymmetric C atom, that is, an atom with 4 different substituents. Most natural monosaccharides have the same configuration at C5 as D-glyceraldehyde, hence, such belong to the D series. The open-chain form of glucose shown here is found in a neutral solution in less than 0.1% of the molecules. The reason for this is an intramolecular reaction in which one of the O groups of the sugar is added to the aldehyde group of the same molecule. The previous intramolecular reaction mentioned gives rise to a cyclic hemiacetal. In aldohexoses, the hydroxy group at C5 reacts preferentially and a 6-membered pyrene ring is formed. Sugars that contain this ring are called pyranoses. By contrast, if the O group at C4 reacts, a 5-part furan ring is formed. In turn, sugars containing this 5-membered ring are called furanoses. In solution, pyranose forms and furanose forms are present in equilibrium with each other, and with the open-chain form. However, in glucose polymers, only the pyranose form occurs. The Haworth projection shown here is usually used to depict sugars in the cyclic form, with the ring being shown in perspective as viewed from above. Depending on the configuration, the substituents of the chiral carbon atoms are then found above or below the ring. OH groups that lie on the right in the Fischer projection appear under the ring level in the Haworth projection. On the other hand, OH groups on the left appear above the ring. As a result of hemiacetal formation, an additional chiral center arises at C1, which can be present in both possible configurations. This is called the anomeric carbon, and the different isomers arising are termed anomers. To emphasize this, the corresponding bonds are shown here using wavy lines. To continue, the Haworth formula does not take account of the fact that the pyrene ring is not plane, but usually has a chair conformation. As shown here, 2 frequent conformations of D-glucopyranose are shown as ball and stick models. In the 1C4 conformation, at the bottom part, most of the O groups appear vertical to the ring level, as in the Haworth projection, and are termed as in an axial or a position. In the slightly more stable 4C1 conformation, as shown in the top, the O groups take the equatorial or E position. At room temperature, each form can change into the other, as well as into other conformations. Let us now proceed to the different forms of isomerism in carbohydrates. As we mentioned in the previous slides, a carbon atom that contains 4 different chemical groups forms an asymmetric or chiral center. The groups attached to the asymmetric carbon atom can be arranged to form 2 different isomers that are mirror images of each other and not superimposable. They are termed as enantiomers. Monosaccharide stereoisomers are designated D or L, based on whether the position of the hydroxyl group furthest from the carbonyl carbon matches that of the D or L glyceraldehyde. The D and L designation is still used in medicine for describing sugars and amino acids. Because glucose, the major sugar in human blood, and most other sugars in human tissues belong to the D series, sugars are assumed to be D unless L is specifically added to the name. To continue, stereoisomers have the same chemical formula, but differ in the position of the hydroxyl group, on one or more of their asymmetric carbons, as shown here. A sugar with n number of asymmetric centers has 2 raised to the n number of stereoisomers unless it has a plane of symmetry. Epimers, on the other hand, are stereoisomers that differ in the position of the hydroxyl group at only one of their asymmetric carbons. D-glucose and D-galactose are epimers of each other, differing only at position 4, and can be interconverted in human cells by enzymes called epimerases. D-mannose and D-glucose are also epimers of each other, differing at position 2. However, mannose and galactose are not epimers, because the hydroxyl positions differ around more than one asymmetric carbon. They are not termed epimers, but diastereoisomers, instead. Monosaccharides exist in solution mainly as ring structures, in which the carbonyl group, either an aldehyde or a ketone, has reacted with a hydroxyl group in the same molecule, to form a 5 or 6-membered ring. The oxygen that was on the hydroxyl group is now part of the ring, and the original carbonyl carbon, which now contains a O group, has become the anomeric carbon atom. A hydroxyl group on the anomeric carbon drawn down below the ring, is in the alpha position. On the other hand, if the O group is drawn up above the ring, it is in the beta position. In the actual 3-dimensional structure, the ring is not planar but usually takes a chair conformation, as we previously discussed. This will assure that all the hydroxyl groups are located at a maximal distance from each other. These corresponding isomeric forms are called anomers. To state it in another convention, in the beta anomer, the O group at C1 and the CH2OH group lie on the same side of the ring. In the alpha anomer, they are on different sides. The reaction that interconverts anomers into each other is known as mutarotation. This concludes this episode of the BiochemSerye series of this topic. Feel free to watch the other BiochemSerye episodes of this lecture as linked on the next screen, and in the description below. Please subscribe to our channel, the BiochemSerye channel, click on the notification bell button, and be notified of new videos to be uploaded. Content will be added regularly. Thank you for watching this BiochemSerye episode. You can check out the related videos linked here or click on the related playlist as shown to view all the videos of this topic.